Hello students, I am Dr. Muhammad Yaseen. Today our lecture is about the introduction and classification of alkaloids. Here is a list of the key concepts that we are going to learn today. We will have first some definitions of alkaloids, then we will discuss their functions in plants and animals. We will also study in this lecture the nomenclature of the alkaloids and different types of methods used to classify the alkaloids. We will also discuss the physical and chemical properties of alkaloids. We will then study the qualitative chemical test used for alkaloids, followed by the methods used for the isolation of alkaloids. At the end of this lecture, we will discuss some quantitative chemical assays for alkaloids. So these are the key concepts that we are going to learn today. The alkaloids are one of the most diverse group of secondary metabolites found in living organisms and have an array of structure types, biosynthetic phase and pharmacological active phase. Although alkaloids have been traditionally isolated from plants, an increasing number are to be found in animals, insects, and marine invertebrates and microorganisms. Many alkaloids have been used for hundreds of years in medicine, and some are still prominent drugs today. Hence, this group of compounds has had great prominence in many fields of scientific endeavor and continues to be of great interest today. Why do plants expend so much of their vital resources on the biosynthesis of alkaloids? How do they produce alkaloids, and what are the mechanisms of regulation of biosynthesis and location within plants will be discussed in the incoming lectures. Alkaloids mean alkali-like substances or basic nitrogenous compounds of plant or animal origin and generally possessing a marked physiological action on men or animals. The nitrogen is contained in a heterocyclic ring system and is mainly derived from amino acids. If we talk about the historical importance of alkaloids, then human recognition of alkaloids is as old as civilization, since substances have been used as drugs, as potions, medicines, teas, poultices, and poisons for 4,000 years. It is likely that in the hunt for food and dealing with enemies, particular use was made of plants containing alkaloids for arrow poisons, and this use probably preceded their medicinal use. Even today, these poisons are still in use in Africa and South America. One very important question comes: why living organisms spend a lot of energy in the synthesis of alkaloids? What is the role of alkaloids in plants and animals? It is now generally accepted that secondary metabolites have a role in the survival of organisms. In plants, these compounds are involved as attractants to ensure pollination and are found to play an important role in plants' interaction with animals and higher and lower plants. Alkaloids are now generally considered to be part of an elaborate system of chemical defense in plants, and indeed the same seems to be true in vertebrates, invertebrates, and microorganisms. Most alkaloids are physiologically active compounds having a variety of toxic effects on animals. Toxic alkaloids are frequently found as part of conspicuous, often violent insect defense system, and these may be synthesized by the insects or acquired as a part of their diet. However, plants sequester alkaloids for use as a passive defense mechanism by acting as feeding deterrents for predating insects. The bitter taste of many alkaloids may be of importance as a feeding deterrent. Some alkaloids like quinine, strychnine, and purcines are extremely bitter. These are, however, little available data on the role of bitterness in plants and animals' relationship, being for the most part based on human preferences, although it has been suggested that the bitterness of alkaloids is a universal feeding deterrent in the plant's foodstuff, as reported by Bates Smith in 1972. The studies of Wink et al. In 1993 indicated that the alkaloids are a universal feeding deterrents in plant food stuff. Other examples of alkaloids as defensive chemicals in animals are to be found in marine ecosystems. Marine sponges may produce toxic alkaloids such as lepidolins as a novel type of bisphenolozidine alkaloid. In general, alkaloids are regarded as a part of the plant's constitutive defense. This means that in certain plants, alkaloids occur in a species-specific and genetically programmed accumulation pattern which remains unaffected by herbivory, microbial attack, and mechanical damage or stress. Alkaloids are phytoalexins and rare, and in most cases evidence for this role is still somewhat controversial. In cell culture derived from the plant, elicitation of the production of alkaloids by various techniques can be produced quite startling, increasing in the yield. Important to the role of alkaloids in chemical defense is the location of alkaloids within the plant. For protection, the alkaloids must be localized at the site to be protected and be at a sufficient concentration to be an adequate defense. Over 100 years ago, botanists noticed that alkaloids, as are other secondary metabolites, are usually found where herbivores' attacks would most affect the plant's fitness. That is, in the fluorescence and young growing tips and in the peripheral cell layers of the stem and root. These findings have been repeatedly confirmed in the intervening years. Alkaloids after the discovery of Morillon in 1819 and Hartmann in 1991. Alkaloids, although apparently present in low levels, that is 0.1 to 2% dry weight, are mostly concentrated in specialized part of the plant and the immediate concentration is much greater. A good example is the concentration of morphine, which in whole poppy capsule is about 0.3 to 1.5%, but in crude opium, the air-dried poppy latex is 25% or more. Alkaloidal plants were also used by hunters, priests, witches, and magicians. Some alkaloids that interfere with the transmission of nerve impulses to the muscles and thus led to immediate paralysis, such as aconitine, which was still in use in medieval Europe. Texas alkaloids, atropine, texaferrine, and tubochlorine played a significant role as arrow poisons for hunting and warfare. The ancient Greek word toxicon originally meant poison for arrows. In Central and South America, frog of genera Phyllobates and Dendrobates secretes toxic alkaloids such as batrochotoxins. Batrochotoxin is 5 times more potent as tetrodotoxin and achieves its toxicity by activating sodium channels, thus distributing neurotransmission. Natives used and still use these toxins as potent arrow poison, and the secretions of one frog provides poison for 50 arrows. Before and during the Middle Ages, Several other alkaloids were used for murder and executions. Infamous are quinine, aconitine, atropine, and strychnine. Certain were employed in this respect as draught for execution. For example, Socrates' executions in ancient Greece with hemlock, which contained quinine. At the time of Roman Empire, belladonna, the source of atropine, has been mixed with food with the purpose of murdering. Cleopatra, the queen of Egypt, used Egyptian henbane that contains hyacinamide for suicidal purposes. In Central and South America, the use of hallucinogenic plants followed a different route. Sources indicate that medicinal and psychotropic plants and mushrooms have been used by Indians for at least 3,000 years. Written evidence originates from the 16th century, for example from the Hernandez. We can speculate that psychotropic plants were first discovered by Indians of lower social status, but that this knowledge was taken over and kept by the shamans, where it has remained exclusively until today. Besides shamans, brujos, witch doctors, or the herbalists, or the healers still exist in modern Mexico, whereas the former two are the most concerned with the magic and divination. The herbalist and the healer, who are lower in social status, collect plants for medicinal application. Although the Central and the South America were Christianized 4 centuries ago, some of the magico-religious rites of the Aztecs still exist today. Here is a list of the key concepts that we are going to learn today. We will have first some definitions of alkaloids, then we will discuss their functions in plants and animals. We will also study in this lecture the nomenclature of the alkaloids and different types of methods used to classify. There is no unique method for naming alkaloids. Many individual names are formed by adding the suffix "-ine" to the species or genus name. For example, atropine is isolated from plant Atropa belladonna. Strychnine is obtained from the seed of strychnine tree. Where several alkaloids are extracted from one plant, their names are often distinguished by variation in the suffix. For example, iodine, I-D-I-N-E, iodine, A9, A-N-I-N-E, A9, and similarly A-line, A-L-I-N-E, A-line, and I9, I-N-I-N-E, I9, etc. There are also at least 86 alkaloids whose names contain the root win, V-I-N, win, because they are extracted from vinca, V-I-N-C-A, vinca plants such as Vinca rosea. These are collectively called as vinca alkaloids. So, there are different methods for the naming of alkaloids. Some alkaloids are named on the basis of the genus from which they are derived or isolated. For example, I had quoted the example of atropine, that is isolated from Atropa. Similarly, the names of some of the plants are named after the species name. For example, cocaine is isolated from coca plant. Some alkaloids have got the common name. For example, ergotamine is derived from ergota. Similarly, the names of some alkaloids are based upon their physiological activity. The example of this name is amidine, derived from amitate. Some alkaloids have got the names after the name of their discoverer. For example, palityrine is derived from the name of the discoverer who discovered it. alkaloid, this alkaloid, and the name of that discoverer was Pelletier. So the Pelletier is derived from the name of Pelletier. So there is no any hard and fast rule for the nomenclature of alkaloids. Some alkaloids have got their name from the genus, some have got from the name of the species from which they were isolated, some have got the common names, some names are based upon their physiological activities, and the others have the names after the name of their discoverer. Now let us discuss some of the most commonly used prefixes that are used to designate a particular structural conditions to an alkaloid. The prefix NOR designates M-demethylation. What is meant by the M-demethylation? M-demethylation that particular alkaloids don't have any alkyl group or methyl group directly attached with the N atom in the structure of that alkaloid. For example, NOR-pseudoephedrine. So if you compare the structure of pseudoephedrine with the NOR-pseudoephedrine, the difference is that in the pseudoephedrine, one methyl group is attached to the nitrogen atom, whereas in the NOR-pseudoephedrine, this methyl group is absent. And similarly, if you compare the structure of NOR-nicotine and nicotine, the difference is that in NOR-nicotine there is no methyl group directly attached with the nitrogen atom, whereas in the nicotine one methyl group is attached with the nitrogen atom. Apo prefix designates dehydration. Let us discuss the structure of apomorphine. If we compare the structure of the morphine with apomorphine, then the structure of apomorphine lacks one water molecule. So if you dehydrate or remove one water molecule from the morphine, then you obtain another alkaloid called apomorphine. Similarly, iso, pseudo, nu, and api prefixes are used to indicate different types of isomers of a same alkaloid. So these iso, pseudo, nu, and api indicate the isomers of the same type of the alkaloid. So if an alkaloid exists in different types of isomers or in the forms of different isomers, they may be designated as iso, pseudo, nu, and api. Hello students, I am Dr. Muhammad Yaseen. Today our lecture is about the introduction and classification of alkaloids. Compared with most other classes of natural compounds, alkaloids are characterized by a great structural diversity. There is no uniform classification. Initially, when knowledge of the chemical structures was lacking, botanical classifications of the source plant was relied on. This classification is now considered absolute. Absolute means it is ruled out. More recent classifications are based on similarity of carbon skeleton, for example, indole, isoquinoline, and pyridine-like, or biochemical precursors such as orthonine, lysine, tyrosine, and tryptophan, etc. However, they require compromises on borderline cases. For example, nicotine contains a pyridine fragment from nicotinamide and pyrrolidine part of the orthonine, and therefore can be assigned to both classes. Alkaloids are often divided into the classified on the basis of pharmacological action or biological activity, or they may also be classified on the basis of their chemical structure, or on the basis of their biochemical origin, or on the taxonomical origin. So people have classified the alkaloids on the basis of their pharmacological action, chemical structure, biochemical origin, and taxonomical origin. Hagner classified the alkaloids into three main types. According to the Hagner's classification, which is based on both the type of the nitrogen and the biochemical origin of the alkaloids can be classified into three classes, namely true alkaloids, proto-alkaloids, and pseudo-alkaloids. True alkaloids are those alkaloids which are derived from the amino acids and have nitrogen in a heterocyclic ring. So the example of a true alkaloid is the atropine and morphine. So any alkaloid that is derived from an amino acid and possess an heterocyclic nitrogen, that is nitrogen in a ring, it is classified as true alkaloid according to the Hagner's classification method. Whereas the second type of alkaloids are called the prototype alkaloids. Prototype alkaloids are also derived from the amino acids and do not have nitrogen in a heterocyclic ring. So they don't have nitrogen in a heterocyclic ring. Their heterocyclic ring may comprises of oxygen or any other than the nitrogen atom. So those alkaloids that don't have nitrogen in their cycle, in their heterocyclic ring, they are called as proto-alkaloids, and these are also derived from the amino acids. So the difference between the true alkaloids and the proto-alkaloids is that these both are derived from the amino acid. However, the true alkaloids have nitrogen in a heterocyclic ring, whereas the prototype alkaloids don't have any nitrogen in their heterocyclic ring. Now let us discuss the pseudo-alkaloids. The third type of alkaloids as classified by Hagner are the pseudo-alkaloids. Pseudo-alkaloids are the alkaloids that are not derived from the amino acids but have nitrogen in a heterocyclic ring. So the pseudo-alkaloids contain a heterocyclic nitrogen atom. They are not derived from the amino acid. However, they are derived from non-amino acid precursors. They are weakly basic in nature. For example, purine alkaloids, caffeine, steroid alkaloids are the examples of pseudo-alkaloids. Pseudo-alkaloids are weakly in basic. If we compare the pseudo-alkaloids with the proto-alkaloids and the true alkaloids are basic in nature, form water-soluble salts. The examples are the quinine, atropine, and morphine. Whereas the examples of the proto-alkaloids include the colchicine and ephedrine. They are also basic in nature. So the summary is that according to the Hagner classification, alkaloids are of three types: true alkaloids, proto-alkaloids, and pseudo-alkaloids. True alkaloids are derived from the amino acid and contain heterocyclic nitrogen, and they are basic in nature. The examples of the true alkaloids are quinine, atropine, and morphine. Whereas the proto-alkaloids contain nitrogen but not in a ring system, so their heterocyclic ring does not comprise of nitrogen. They are also derived from amino acids, sometimes considered as the biological amines. So the proto-alkaloids are also called as the biological amines. The examples are the colchicine and ephedrine. These are also basic in nature. Whereas the pseudo-alkaloids contain a heterocyclic nitrogen atom. They are not derived from amino acid. However, they are derived from non-amino acid precursors and are weakly in basic. Examples are the purine alkaloids such as caffeine and steroid alkaloids such as solanidine. Based upon their biological activity or pharmacological activity, alkaloids may be classified as analgesics, central nervous system stimulants, anti-cancer, mydriatics, myotics, anti-asthmatics, antitussive, expectorants, antihypertensive, smooth muscle relaxant alkaloid, skeletal muscle relaxant alkaloid, anti-myotics alkaloids, and anti-parasitics alkaloids. Analgesic examples of the analgesic alkaloids include morphine and codeine. Analgesic alkaloids are used for the relief of pain. The caffeine alkaloid is found in coffee and in tea. That's why when someone takes or drinks tea or coffee, he or she feels active because caffeine activates the central nervous system. Example of the anti-cancer alkaloids include vincristine and vinblastine and taxol. Mydriatics are the alkaloids that dilate the pupil, and a blurred eye vision is the result of the mydriatic drug. So the side effect of mydriatic drugs is the blurred eye vision. Example of the mydriatic drug alkaloid include atropine. Myotics are the drugs that contract the pupil. Example of the myotic drug is pilocarpine. Anti-asthmatic alkaloids include ephedrine. Anti-asthmatic alkaloid or ephedrine is used for the relief of asthma. Similarly, expectorants include, the example of the expectorant include lobeline. Expectorants are used to discharge the sputum from the lungs. Similarly, the example of the antihypertensive alkaloids include razapine. There is another method used to classify the alkaloids, and it is named as chemical method of classification of alkaloids. In the chemical method of classification of alkaloids, alkaloids are classified on the basis of their chemical structure. We will discuss this method of classification in the part 2 of this lecture. At the end of this lecture, here it is a list of the recommended books. You may consult any one of them.